Today we're going to have a look at the special senses, and in this session we're going to look at the vision and the role of the eye in vision. So really at the end of the session we want you to be able to describe the basic structure of the eye and its associated structures, but also the function. So how does vision work? Which pathways does it use? What's the difference between rods and cones, for example? So how does the eye actually see? Because the eye can only perceive a very small spectrum of electromagnetic waves. And in order to see an object, of course, the pattern of the object, that must fall on the vision receptors. So that's your rods and your cones in the retina. And they have to be accommodated. And accommodation really is just the process of adjusting the shape of the lens so that the external image falls exactly onto the retina. So basically so that it's in focus. The amount of light entering the eye must be regulated. Too much light will bleach out the signals. The energy from the waves of photons must be transduced into electrical signals, and that's what the rods and the cones do. They absorb that light, and that light is then transduced into electrical signals, into graded potentials and then action potentials, so that we can interpret that information through the visual cortex. And the brain, as I've just said, must receive and interpret those signals. So if we take a really crude look at the anatomy of the eye, the front of the eye is composed of the lens, and that changes shape to focus light signals onto the back of the eye, onto the retina. The retina captures that light in signals and transmits an image to the brain via the optic nerve. And the interesting bit, you can take a look at this, and you can see that at the optic nerve, you're actually at the process where the optic nerve is coming out, you don't actually have any cones or rods. You don't have any photoreceptors. So the brain actually has to make its own images. It basically does some processing to make it seem as though there's an image there. And the center of the retina has an area called the macula, and the light passes to the macula without having to pass through the retinal layers first. And that basically enhances that visual detail. So this is the macula, and you can see this little white region all the way around. This has got a variety of nerve cells in it that we'll talk about later, so bipolar cells and ganglion cells. And the light is coming in this way, and usually it has to kind of make its way through those nerve cells first. But at the macula, where we have the greatest visual detail, they've been pushed off to the sides. The light can get straight through to that area. So therefore, acuity is the ability to resolve that detail. And the macula is therefore required for focusing during activities such as reading, for example. Now, when we're thinking back to the lens, and we've all heard about the lens, and when the lens gets clouded, that's when you get cataracts. And that causes the world to appear distorted, as though it's through frosted glass, for example. And that's often due to age-related hardening and thickening of the lens. It can also occur in diabetes mellitus, or people who are heavy smokers, or through frequent exposure to bright sunlight. The iris that you can also see there, that helps to contract the different muscles to allow more or less light to enter into the eye. So in other words, it's contracting and dilating the pupils. So here are some of the accessory structures of the eye. And what I'd like you to do is taking a little bit of a look at this. We can see, you've got your eyebrows, your eyelashes, your eyelids, the sclera, which is covered in conjunctiva, your iris and your pupil. But what I really want you to think about, what is the function of the eyebrows and the lashes? What's the function of the eyelids? And what are the functions of the lacrimal glands and the ducts? So I'd like you to just press pause and just think, what is it? What are the key features? What are the key features of each bit? So your eyebrows and your lashes, well, of course, they help to protect the eyes from excess light, from sweat, and from dust. The eyelids, well, they help to spread tears. They shade the eyes for sleep, and they're involved in protection as well. And then you've got the lacrimal glands and the ducts. Well, these are the things that produce tears. And in your tears, you've got water, you've got salts, you've got mucus, you've got immunoglobulins to help your immune system to fight any bacteria or viruses that may get there. You've got lysozyme there, and lysozyme is antibacterial. And so as you might expect, the blinking process with the eyelids, that is basically spreading the tears, the lacrimal secretions. But when the lacrimal fluid secretion increases substantially, then the tears spill over the eyelids. So they spill over the eyelids and fill the nasal cavities, causing congestion and the sniffles that sometimes you get. So it occurs when the eyes are irritated or when you're emotionally upset, for example. And with eye irritation, this helps to wash away the irritant, so it's a very useful process. But with emotional tears, the importance of tears is really very poorly understood. Now, we talked about the conjunctiva there as well, and the conjunctiva is just a transparent mucous membrane. It only covers the white of the eye, not the cornea, and that's the clear window over the iris and the pupil. And it helps to produce that lubricating mucus to help to prevent the eyes from drying out. So if we're looking at the anatomy of the eye and taking a look at the structure of the eyeball from outermost to innermost. So the first thing that you can see is the sclera. So there's your scleral layer. That's white and opaque, helps to protect and shape the eyeball, and it helps to anchor the extrinsic eye muscles, which are there to the eyeball itself. You've got the cornea, okay, and so if you're looking there, connected through, you've got the sclera moving through to the cornea, and this bulges out from a junction with the sclera, and the light rays pass to the interior of the eye. And actually, in the cornea, you have many pain receptors. You've then got the choroid. So again, there we go. There's your choroid. That's got your blood vessels supplying the various eye layers. And so basically, it's helping to nourish those eye layers. And its brown pigment helps to absorb light, preventing it from reflecting and scattering within the eye, which would obviously cause visual confusion. And then you've got the ciliary body, which is shown here. And this is where the choroid basically starts to become the ciliary body, and that's made up of your smooth muscle. So you can see the choroid basically flowing into the ciliary body, and that has the smooth muscle. And then we look at the iris. So the iris, this is the visible coloured part of the eye. And again, it contains smooth muscle to help to dilate or constrict the pupil. So, for example, if you have bright lights and close objects, the pupil constricts. The sympathetic fibres help to control pupillary dilation, and parasympathetic fibres control constriction. But although the iris actually comes in different colours, they do only contain one brown pigment, which is melanin. But when there's a lot of pigment, they appear brown or a darker brown, and a small amount of pigment at the posterior of the iris leads to the unpigmented part scattering the short wavelength light to make the eyes appear green, blue, or grey. And interestingly, most newborn babies have grey or blue eyes, as their iris pigment is not yet fully developed. You've got the retina at the back, and we've talked about that a little bit. That contains the rod and the cones, so those are the photoreceptors. We've got the lens, and that's adjustable to help focus the apparatus. And again, we briefly touched on that as well. And you've got the humours as well. So you've got the vitreous humour and the aqueous humour. And the vitreous humour helps to maintain the shape, and the aqueous humour helps to provide nutrients to the lens and to the cornea. So just quickly going on to a little bit of anatomy and how we control pupil dilation and pupil constriction. And it's done under autonomic control. So if we're looking at the iris, in particular, you've got two muscle groups. You've got the sphincter pupillae and the dilator pupillae. And if you have parasympathetic stimulation, that's during rest and digest, remember, the sphincter pupillae contracts, and therefore you get, and as you can see, as it contracts down, you get the pupil size decreases and less light is coming in. And that's great for reading, for example. And then you've got sympathetic activation, and this is where the dilator pupillae muscle contracts and the pupil size increases. This allows more light into the eye, and therefore you get greater detail, which is what you might want during a fight or flight response. So what is accommodation? So we know accommodation is the process of adjusting the shape of the lens so that the external image falls exactly onto the retina. The cornea does most of the focusing, and the lens just provides the fine-tuning. So if we're looking at what's actually happening, if we're looking at this nice little tall daisy, the light comes in, hits the cornea, and it does a lot of focusing, so a lot of bending of light, hits the eye lens, and that's providing the fine-tuning. And you can also see, if you're looking how the image is actually upside down compared to the original image, if you want to think of it like that. But why do you get this bending? Why do you get this refraction? Well, it's just down to the alteration of the speed of light being transmitted. The light speeds up as it passes into a less dense medium, and slows down as it passes into a denser medium. So basically the light is bent 3 times. The light is bent when it enters the cornea. The light is bent when it enters the lens, and it's bent again when it leaves the lens. But like I said, the cornea accounts for the majority of the bending power. However, the refractory power of the cornea is constant. The ciliary muscles, they contract and relax and help to change the shape of the lens. And the thicker the lens, the more the light bends. So you can't see something close and far with both objects in focus at the same time. So as you can't do this, what I'd like you to do is just hold out your thumb about a foot away from your eye and look at it. Now alternately focus on the thumb and something far in the distance, so that's right above your thumb. And you'll notice that you can't be able to focus at both of them at the same time. And the closest someone with normal vision can actually focus on is about 10 centimeters or 4 inches. And you can try this by bringing your thumb or your finger in closer and closer. And the closest you can really focus on is that 10 centimeter area. And that represents the maximum amount that the lens can actually bulge. It is slightly closer in children, and it gradually recedes with age. And it helps to explain why children often will hold their books very close to their eyes when they're reading, and the elderly will hold the newspaper at arm's length, because the lens loses that elasticity with age. And as I've already said, the image formed is inverted, and it's also reversed from left to right. So if you take a look at how this looks, you can see on the back of the retina, it's also smaller, as you'd expect, but it's also inverted, and it's also reversed left to right. What about a couple of little focusing abnormalities? So visual problems, they're related to visual problems that are related to refraction can be due to hypo or hyper-refractive lens or eyeball structural abnormalities. So a normal eye, as you can see, the focal point comes in and it's directly onto the retina. So you can see you've got perfect focusing. You then have nearsighted or farsighted, and you have in your myopic eye, you have short-sightedness. And this terminology really refers to the aspect of vision that is unimpaired. For in other words, close objects are fine, but far objects are blurred. And a concave lens can then be used to help focus properly, or laser surgery can help to flatten the cornea slightly. And it normally occurs because the eyeball is just a little bit too long. You've got then the hyperopic eye, which is your far-sighted, which, as you might expect, you can't focus on close objects, and usually because the eyeball is a little bit too short. And again, if you're comparing those images, you can see it's basically squashed in a little bit, whereas the myopic eye is kind of stretched out a little bit, if we want to think about it like that. And you've got astigmatism, and that's where you have unequal curvatures in the cornea, and the light rays are unevenly refracted, leading to those blurry images. And that can be treated with laser surgery, for example. So we have a couple of questions for you to think about at this stage. So what I'd like you to do is just press pause, have a little bit of a think about these questions, and just to give you a clue, there's only one correct answer for both of these questions. Okay, you plan to go walking in the mountains, and before you arrive, you can see the mountains clearly in the distance. However, you can't focus on a close object at the same time. Why is this the case? The answer clearly is answer 4, because your eye does not have enough depth of field. Number 2, in order to focus on close objects, your eye lens needs to bulge. So hopefully you got that. That's the first one. So we'll just do a little bit on the physiology of the eye, and then we'll go back to structure again before we do a bit more physiology. Now, the retina consists of millions and millions of photoreceptors. And so when the light is coming in, so here's your pathway of your eye. So take a little look at this image of the eye. You can see the pathway of the light coming in, and there's your retina at the back. And this is just a bigger image of it. And so just so you've got it into context where we are, we've got the sclera there. You've got the choroid, which is this bit, the blood vessels, remember the choroid. Then you've got your rods and cones, which are there, which is shown in yellow in the other image. And then you've got your plexiform layer. Okay, so that's your nerve cell layer. And so as you can see, for most of the part of the retina, the retinal layers, you can see that light that's coming in has to get through the nerve layers before it gets to the photoreceptors themselves. And obviously that can fragment or distort the light slightly. So the choroid, as we've talked about, that helps to provide the blood and the nutrients. You've got the photoreceptors, the rods and the cones. They absorb the light and convert that to an electrical signal. And you've got the nerve cells, which help to transfer the signal from the rods and cones to the brain for interpretation. And vitamin A is needed by the photoreceptor cells, and it's stored in the outer pigmented retinal layer. And like we said, the inner neural layer has millions of photoreceptors and that direct role in vision. And the optic disc that you can also see there, that little region there, this is where the site where the optic nerve leaves the eye. And as you can see, it lacks any photoreceptors. So it's also called your blind spot. And so the focused light on it, so the light that's coming in at all these areas, it can get to its relevant rods and cones. But in this optic disc, there are no rods and cones there. So it's your blind spot. So the light focus on that particular spot can't be seen. However, we don't notice that. We don't see those gaps as the brain uses a very sophisticated process called filling in to deal with the absence of input, and it basically predicts what should be in place in that blind spot so we don't see it as something abnormal or wrong. So we've done a little bit on this already. So the inner neural layer is composed of three main neuron types. The light is coming in from left to right, and you can see it has to get through these nerve fibers first. So you can see there are optic nerves in the way. You can see with the ganglion cells. You can see your bipolar cells, and you've got a variety of other types of cells as well, such as your horizontal cells that are kind of communicating between the rods and the cones and adjusting signals. And here we've got our photoreceptors. So the rods you can see there, and you've got the cones, which are the color ones because they're nice and green and red and blue, and hopefully you can see all of that. And so those three types of cells, we said, the photoreceptors, the bipolar cells, and the ganglion cells. So there are other cell types there that are helping to process the information as well. The signals spread from the photoreceptors through the bipolar cells and on to the ganglion cells. And the bipolar cells communicate via graded potentials. And the ganglion cells, as you can see, they make a right angle turn at the inner face of the retina and leave the posterior aspect of the eye as the thick optic nerve. And about 70% of all sensory receptors are in the eye. There are around about 250 million sensory receptors in the eye. So it takes a huge amount of processing power. And action potentials are generated in the ganglion cells, which then exit the eye as fibers of the optic nerve, as I've already said. So what about these photoreceptors and phototransduction? Well, I've already said that the rods and cones, they're basically modified neurons. And the molecule changes shape as they absorb the light at the appropriate frequency that's coming in. The cones, they're very low sensitivity, so they need bright light for activation. And they have one of three pigments for colored vision, so your reds, your greens, and your blues. And if we're looking at the eye again, you have the fovea centralis. So this is a tiny pit in the center of the macula, and that contains only cones, and that's where your best vision is. So you remember I said at the macula, that's where the other nerve cells disappear, so the light can come in and just basically not get distracted by these other layers. But right in the center of that macula is this tiny pit called the fovea. And so if we're taking a little bit of a look at this, so this is the number of photoreceptors per millimeters squared, and this is basically the angle if we're comparing it to the eye and light potentially coming in. So you can see where the optic disc is, and you can see there are no rods or cones in that area, and that's where you have that filling in process going on. And you can see that going off to that side, so going off this side, you can see we've pretty much only got rods there, okay, very low number of cones. And you can also see going the other way, you've mainly got rods away, apart from in the macula, and in particular right in the middle of that, the fovea, where you've got a huge number of cones, and that's where you can get your color vision, and that's where you get that best vision, that best acuity. And color blindness is due to the lack of one or more of those cone pigments. So, rods are much more numerous than the cones. They're mainly found away from the fovea. If you remember, in the fovea, we've got loads and loads of cones for that best vision. And so therefore, they're involved more in that peripheral vision. They're very sensitive and they respond to very dim light. So therefore, they're more sensitive to light than the cones. The cones, if you think about it, when it's getting quite dark, you really can't see color very well. And that's because there isn't enough light to have that color vision. So you see mainly black and white as it gets darker and darker, or grayscales rather. The rods are best suited for night vision, and they contain that single pigment. So there's no color vision or sharp images, and you get these vague gray tones. And the rods, they respond to almost any photon, whilst the cones, as we've said, have specific ranges of specificity. So how do the photoreceptors actually translate the incoming signals into electric signals? So we have vitamin A, and we all know vitamin A is required for good vision. And remembering, of course, that different photoreceptors absorb light at different wavelengths. So vitamin A and retinal, that basically binds with opsin to form this molecule, and it's very simplified, and you don't need any more detail than this. But we get this molecule called rhodopsin, okay, made up of retinal and opsin. And it's the retinal portion of the rhodopsin that absorbs the light and goes on to change shape. So the rhodopsin absorbs the light, we get a conformational change, they kind of separate, and we initiate an enzymatic cascade. To keep a long story short, we get an enzymatic cascade is initiated, and the photoreceptors hyperpolarize. So in other words, they become more negative. So normally when we stimulate something, we think about depolarization to therefore help to generate an action potential. But in this case, we actually hyperpolarize. So how do photoreceptors translate incoming signals into electrical signals? So here is this diagram again that we've talked about. The light is coming in, hitting the ganglion cells, and they're going through the ganglion cells and the bipolar onto the photoreceptor cells. The light is absorbed by these photoreceptors, so these rods and the cones. We get a conformational change, and the photoreceptors hyperpolarize. And this is shown in this nice little image. You give a nice little light flash, and you can see the membrane potential becomes more negative in these photoreceptors. So we're hyperpolarizing. And therefore, the photoreceptors are inhibited on light absorption and release less inhibitory neurotransmitter. So in other words, there's less inhibition. And the receptor that's the neurotransmitter is actually glutamate in this case, and it's acting as an inhibitory neurotransmitter. It usually acts as a stimulatory one, but in this case, it's acting as an inhibitory neurotransmitter. So if we're really thinking about what's happening, in essence, photoreceptors in the retina are continuously undergoing tonic activity. In other words, they are always slightly active, even when they're not being stimulated by light. And in neurons that exhibit this tonic activity, the absence of a stimulus maintains a firing rate at a baseline. And so while some stimuli can increase that firing rate from the baseline, other stimuli can decrease it. So what's happening in the absence of light? In the absence of light, these bipolar cells that we've been talking about, so these are the kind of gray, the kind of purpley blue ones. In the absence of light, these bipolar neurons that connect the rods and the cones to the ganglion cells outside them are continuously and actively inhibited by the rods and cones by that neurotransmitter glutamate. Remember, it's acting as an inhibitory neurotransmitter as opposed to its usual role as a stimulatory one. So when the photoreceptors now become hyperpolarized on activation by light, they basically switch off the release of glutamate. They're becoming hyperpolarized, so they release less glutamate. And with less glutamate being released from these bipolar cells, from the photoreceptors, the bipolar cells is now becoming more depolarized in the light situation, but the photoreceptor is hyperpolarized, if that makes sense. Just that, and we'll go through it again in a second. I think it's on the next slide as well. I just want to quickly mention some of these other cell types, and you really do not need to know the detail of this because it's really quite complex. But there's a large degree of processing of visual information occurs within the retina itself before visual information is even sent to the brain. There are many other cell types involved in moderating the signal. For example, sometimes these horizontal cells carry signals from one rod or cone to other photoreceptors and to several bipolar cells. And when a rod or cone stimulates a horizontal cell, this horizontal cell can inhibit more distant photoreceptors and bipolar cells, therefore creating lateral inhibition. And this inhibition actually helps to sharpen the edges and enhances the contrast in images by making regions receiving light appear lighter and dark surroundings appear darker. So it's quite complex, but they have a very important role to make that image a lot clearer. So how is hyperpolarization transmitted to the brain? So remember what we said. So the initial signal, it comes in, we're activating these we're stimulating these photoreceptors. But instead of getting depolarization, we're getting hyperpolarization of the photoreceptors cells. Now they're normally releasing glutamate, but now they release less glutamate. And this glutamate is usually acting inhibitory onto the bipolar cells, but we're getting less inhibition at this point now. So the bipolar cells, which are no longer inhibited, now can release excitatory neurotransmitter onto these ganglion cells. And as it happens, its glutamate being released from the bipolar cells is now acting as an excitatory neurotransmitter onto the ganglion cells. And the ganglion cells can now generate action potentials, which are transmitted in the optic nerve, so cranial nerve 2, that goes to the brain. So what is the visual pathway to the brain? So again, this is very much simplified for you. So the optic nerve is now taking that signal. The optic fibers cross at the optic chiasm, and that information goes to the thalamus, onto the primary visual cortex, and onto the visual association area, and that basically is giving you that information about what the image is and how to interpret it, that higher knowledge base, if you like. And so we're getting the understanding of what we're seeing. It also goes to the hypothalamus, and the specific region in the hypothalamus it goes to is the hypothalamic suprachiasmic nucleus, if you want to know that. And that helps us control circadian rhythm. So in other words, it's involved in our internal clock, involved in sleep and alertness cycles. And then it also goes through the brainstem, and in this case it's the superior colliculus, and this is a reflex, to the visual reflex center, where if a bright light suddenly comes at your eyes, you turn your eyes and your head away in response to that bright light. So that's your reflex. So if you suddenly get a really bright light flashing in front of you, you turn your eyes and your head away, and that's going through the superior colliculus. So just a couple of review questions for you to think about when we're finished. You really don't need to know that much more physiology than that. That level of physiology is fine. It is much, much more complex than that. So just if you understand that, that's good enough for us. So here are your review questions. Press pause, have a bit of a think, and then I'll go through it. So if light rays are to be focused onto the retina, then the closer the object is to the eye, the curvature must be, permitting a something degree of accommodation. Which is the correct answer? Hopefully you got its number 1. So if light rays are to be focused onto the retina, then the closer the object is to the eye, the greater the curvature must be, permitting a greater degree of accommodation. Number 2, the second question. Now, more than one of these is correct. Which of the following are correct concerning the image that falls on the light-sensitive part of the eye? The image is reduced in size. Correct. If you're looking at a car, the car is clearly smaller on the eye, but on the retina. Right side up. That is incorrect. It is reversed. And smaller as the distance between the object and eye is increased. That is also correct. So it becomes smaller as the distance between the object and the eye is increased.